So what do lasers, ultrasound, and cancer have to do with each other? This project might someday diagnose cancer using ultrasound and laser light. Meet the box. Outside the box is a laser. This one uses helium and neon to make a red beam. The beam comes out here, goes through a polarizer, travels over here and hits a mirror, goes through another polarizer, and then travels into our box. Now we're inside the box and we can really see what's going on. Normally this would be filled with water, but it's drained so we can see the mechanism more clearly. We use this syringe to inject our sample into our testing chamber. This mechanical injector makes sure it injects at a steady rate. This is the path that the cells or whatever we're testing will travel into our machine. It comes out here, down at the bottom of this really thin tube. Then it's sucked away in this funnel. This whole injection system is called a flow cytometer. Its job is to make the cells or other particles we're testing flow out one at a time at the bottom of that narrow nozzle. The funnel collects the cells after testing so they don't get in the way. Inside the testing chamber is also an ultrasound transducer that sends out a pulse of sound into the stream of cells, causing them to jiggle. So why are we causing them to jiggle? Cancer cells have a different internal structure than normal cells, and they'll vibrate differently, just like different bells. This will change the pattern of light we see in our detector, so we should be able to tell the difference between the two. So let's review the setup of our experiment. It starts with a laser coming into the chamber through a small window. That laser comes in and then passes through a stream of cells. A pulse of ultrasound also passes through the same stream of cells. Everything is at a right angle to everything else. Here is a diagram from the top of our experiment. The black dot represents the stream of single injected cells. As the laser hits the cells, it gets deflected and measured by a detector. We can see the particle as a blip in the signal of the detector. When the ultrasound is on, the cell will vibrate and scatter light differently. We can use the different pattern at the detector to find out exactly how the cell is vibrating. Here the cell bends the laser light, which causes it to hit the detector and give us a signal. Ultrasound moves the cell, the light moves on and off of the detector. This gives a wave pattern for the particle. So wait, what does this have to do with cancer? Just like these springs, a different vibration means a different structure or stiffness. In theory, we can use this difference to tell us something about these cells, and whether or not they're cancerous. Right now, our main problem is getting everything to line up, because ideally, we really want a good signal-to-noise ratio, so everything has to work out perfectly. With a little planning and some good luck, we can get some good data, which is what we need to figure out how to make this work.